Okay, today I'm going to talk to you about using a light microscope. Here is an image of a light microscope. What we use microscopes for is to be able to view tiny things. It magnifies them so we could see them in much more detail, or even see things that we wouldn't otherwise be able to see with the naked eye. They're different from telescopes because telescopes are used to view really distant things, such as planets or stars. Microscopes are used to look at things that are microscopic, very small. So, before we get on to actually using the microscope, if we want to view something with a microscope, first of all we have to prepare it by putting it onto a slide. Now, all a slide is it's a bit of glass that our sample sits on. It's important that it's glass because it needs to be transparent. So let's say I have a thin slice of onion, really thin slice of onion, and I've put it on my slide. Now, what I might need for different, it depends on what I'm looking at, but I might need something called a stain. So all a stain is a liquid that you can drop onto your sample, and it kind of stains different parts of the cell. For example, if you have a stain called methyl blue, it stains the nuclei. So that's all, nuclei, remember, is just the plural of nucleus. It stains all the nuclei so you can see them much better. So after you've put the stain on there, you put on this little square of glass called a cover slip, just to keep everything in place. So that cover slip goes over the top there, and you just need to press it down to get rid of any air bubbles as well. So then this is ready. Your specimen is now ready to be viewed on the microscope. So it's on that slide, so it goes on here. Now this particular part here, where your specimen goes, you can actually clip it down with these clips. This part here is called the stage. So this is where your specimen actually sits, right here. Now it's really important as well that we turn the microscope on, because this part here at the bottom, this part here is the light. So it's important that we have light shining up through the sample like this, so we can actually view the sample itself, okay? The specimen. Right, the other things that we need to know. The bit that you look down, this part here, is called the eyepiece lens. Nice and simply, it goes with your eye. So this here is the eyepiece lens that you look down. The name of the other lenses here are the objective lenses. Can you see there's several different objective lenses? All of them have different strengths of magnification. Some of them have a lower magnification, some have a higher magnification. And also this part here, this is called the focusing wheel. So you turn this in order to bring the image into focus. Now, what you need to do to begin with, once you've set up your microscope, you've got your specimen ready, it's on the stage, you start off with the stage low. So you have it as low as it goes to begin with. Then what you do is you choose the objective lens with the lowest magnification, the weakest magnification out of all of these. What you then do is when that's all set up, and remember your light is on, whilst looking down the eyepiece lens, you turn the focusing wheel, bringing up the stage until it comes into focus. So that means you can see it clearly. It's no longer blurry. What you can then do is a lot of microscopes actually have a fine focusing wheel as well. So the bigger one here, this is the kind of coarse focusing to get it pretty much roughly in focus. And then this smaller one here is to get it perfectly focused. Now, once you've got the image in focus, you can then change the magnification by changing the objective lens. So this part here just swivels round, and you could pick whichever lens you want to pick. After you've swiveled round and chose the lens, it might need some more fine focusing in order to get it in focus. But that's essentially it. That is how to use a light microscope. So just to recap, you need to put your specimen on a slide. You might need to stain it so we can see it better under the microscope, and you put on a little thing called a cover slip, just a bit of glass that goes over the top. That specimen goes onto the stage, which is this part here of the microscope. We always start with the stage down, then whilst looking through the eyepiece lens, we slowly bring the stage up until it's in focus. Then we always start with the weakest magnification objective lens. Once it's in focus, that's when we can change the strength of the objective lens. And that's how to use a light microscope. Thank you.